Hello friends, welcome to our channel. In this video, I will tell you about the chemistry and the physiology of the insulin and its action. So before proceeding, if you are new to our channel and have not subscribed our channel yet, please click on the subscribe button below and do subscribe and share our channel. So let us start with the video. So as an insulin, the topic we are going to discuss, we should have an introduction of this whole topic, what we are going to study. So we are going to discuss about the general introduction, its synthesis, regulation, action, and last the clinical, that's the diabetes mellitus. We first start with the topic of the introduction of the topic, that is the insulin. So the insulin contains 7 characters. So we have those 7 points in the introduction part. First, why the insulin topic so much important from the examination point of view as well as in the applied aspect of the biochemistry and physiology. See, it was the first hormone to be isolated, produced and purified. It was also the first enzyme or the protein to be produced by the recombinant DNA technology. Moreover, it is also the first enzyme to be sequenced. That's why the topic or this enzyme insulin is so much importance. So how it is the enzyme is synthesized from where and how it is synthesized, that we want to discuss. See, it is synthesized by islet of Langerhans, which is part of pancreas. I think all of you might be knowing about this. The pancreas contains 2 parts. It has islet of Langerhans and the other part is the SNI part or the alveoli part. SNI or alveoli. Islet of Langerhans is concerned with the endocrine function and the SNI and the alveoli is concerned with the exocrine function. Islet of Langerhans contains various types of cells, that are the A cells, B cells, D cells and PP cells, that are pancreatic polypeptide cells. A cells are also called alpha cells, B cell is also called beta cell and D cell also called delta cell. See, insulin is secreted by the B cells or the beta cells of islet of Langerhans. So this was about the synthesis or how it is produced by the pancreas. It is an anabolic hormone. That means it helps in the building of the body. This point we are going to discuss in detail of the action of the insulin hormone. Next point is related to biochemistry. That's what is the plasma level of insulin. So it is very important. In the clinical cases, we will be discussing about why the plasma level is so much important of insulin and when it fluctuates and what are the normal range. So the normal range of plasma or the plasma level of the insulin is 2 to 20 micro unit per ml. So this you have to remember. Half-life of insulin means of how much time the insulin concentration will be reduced to half, that is around 5 minutes. It is, as you might be knowing, that insulin is a peptide hormone. Peptide hormone means it consists of a chains of amino acid linked to each other by the peptide bond. Usually I don't tell about the number of amino acids in the peptide bond, but in the case of insulin it is very important. It consists of 51 amino acid. I think everybody might be knowing from the junior classes also, during the preparation time also you might have studied that insulin consists of 2 peptide bonds, A, B, which are joined to each other by C peptide and disulfide bond. Okay, that will be discussing about the synthesis of topic insulin, that we will be discussing. But meanwhile you should know A peptide or the A chain consists of around 20 amino acid and B chain consists around 31 amino acids. So this is very important in the case of the insulin to remember the number of amino acids in the peptide bond. Because as I told you, it was the first hormone to be isolated, produced by recombinant DNA technology, and one more point to add, it is the first hormone to be sequenced. And the 7th and the last point to remember is about the clinical of the topic, that is the insulin, is its deficiency causes diabetes mellitus. Not only deficiency, its inefficiencies can also cause the diabetes mellitus, that we will discuss in the topic diabetes mellitus. It will be a separate topic and a new video I will be uploading soon. So just stay tuned and press the notification bell below. Now let's see the next topic, that is the synthesis of insulin. How insulin is synthesized. See, most of the hormones are synthesized by the ribosome present on the endoplasmic reticulum, and same is the case of the insulin. Insulin is secreted in the form of proinsulin produced by the ribosomes in the endoplasmic reticulum. See, it consists of a chain, it's B chain, C chain and this is A chain. And what is this over here? See, this part is called the signal sequence. signal sequence. So this purple color part is the signal sequence, this pink color are the B and A chain, and this orange color is the C chain. A and B are joined by the disulfide bonds and forms the proinsulin. So pre-proinsulin is converted into proinsulin, and all these changes take place in the endoplasmic reticulum. So endoplasmic reticulum converts the pre-proinsulin into proinsulin. Moreover, the signal sequence function is to direct this pre-proinsulin to the endoplasmic reticulum. So later it is removed. So this is the signal sequence which after processing is removed. So this proinsulin is functionally not active. So it is directed to Golgi bodies, where it is converted into or broken down into separate C peptide or C chain and the insulin part. So let me ask you a question: which among these two chains are the B and A chain? So just look at this length. As I told you, 31 amino acids are present in the B chain. So this will be the B chain because it is longer. This is B chain and this is A chain, which are joined to each other by disulfide bond. Moreover, intermolecular disulfide bond is present in A and B, and intramolecular disulfide bond is present in the A chain. So its synthesis takes place in the endoplasmic reticulum on the ribosomes, which are converted into, so it is synthesized in the form of pre-proinsulin, converted into proinsulin, which is further broken down into Golgi bodies and processed into a separate insulin, and the C peptide, which is that is not metabolically active or function not active, that is the C peptide. Now let's see about the regulation of the insulin secretion, or what factors lead to the secretion of the insulin. So there will maybe some factors that promote the synthesis of insulin, and there might be some factors which decrease the synthesis of insulin. So let's see which factors promote insulin synthesis, and later we'll discuss which factor inhibit insulin synthesis. So in the promotion part, we have got three majority. We have got three things. That's the glucose. Second is the amino acids. Third is the GIT hormones. The topic of the glucose tolerance test, I told you that glucose level, as we take with the food, it rises till up to 2 hours, then it decreases, because from the point of taking of food till 2 hours, the level of insulin decreases, it decreases the level of glucose. So there might be some connection between the GIT and the synthesis of insulin. So that's that is regulated by the GIT hormones. Now first discussed about in detail about how glucose regulate the synthesis of the insulin. The detail of it we will be discussing separately when discussing about the diabetes mellitus and other topics related to the metabolism and the glucose hemostasis, that we'll be discussing separately. But meanwhile, you should remember glucose is the most potent stimulator for synthesis of insulin. And among the amino acid, if you take a protein rich diet, the level of insulin also rises. It is because of majority 2 amino acid, that are the lysine, arginine. Sometimes one more amino acid is also considered, that is the leucine. But the most potent among this is the arginine. So if somebody asks which amino acid affects most the synthesis of insulin, you should mark your answer as arginine. Next priority should be leucine, then lysine. Among the GIT hormones, the most important 3 are the secretin, gastrin, and CCK, that's cholecystokinin. These 3 hormones regulate the insulin synthesis. These functions we will discuss in a separate video on the GIT hormones and their action, that will be uploaded soon. So do subscribe and share channel, and if you like the video, please hit the like button. Now let's see about the factors which inhibit the synthesis of insulin. The most potent or the most important factor is epinephrine. So just keep in your mind that epinephrine decreases the synthesis of insulin from the pancreas. And in the pancreas also, the part which secretes the insulin is the islet of Langerhans. And in the islet of Langerhans, the type of cell is the B cells or the beta cells of the islet of Langerhans. So these are the factors that regulate the synthesis of insulin. We have three factors that regulate the or promote the synthesis of insulin, and the most important the factor which inhibit is the epinephrine. Autonomic nerves, autonomic nerves which supply the pancreas can also, when stimulated, they can also increase the synthesis of insulin. So we have got four factors in the part of the promotion of the insulin secretion, and one in the that inhibit the synthesis of insulin. It does not mean there are not many other factors. There are other endocrine hormones which also inhibit or regulate or promote the synthesis of insulin, but that we'll be discussing separately in the glucose homeostasis, and that the hormones which are antagonistic to insulin, and the other factors that we will be discussing later. Next topic to discuss is the mechanism of action of insulin. So insulin act on some cells by the help of some receptors called insulin receptors. Insulin receptor. Insulin receptor is a tetramer. Tetramer means it consists of 4 units. The 4 units are 2 alpha and 2 beta. It is similar to the hemoglobin, which also consists of 4 subunits. So hemoglobin is also tetramer consisting of 4 subunits, that are the 2 alpha and 2 beta, and same is the case of the insulin. So it is also tetramer. We have 2 alpha chain and 2 beta unit. So we have 2 alpha unit and 2 beta unit. So alpha unit is present outside the cell membrane. So suppose this is the cell membrane. The alpha unit is present outside the cell membrane, and beta chain or the beta unit is partially inside and partially outside, and it is coupled or linked to the alpha unit. The insulin receptors, that the part of the insulin receptor which receives the insulin is the alpha receptors. Suppose this is the insulin molecule, it binds to the alpha receptor, which causes some conformational change in the alpha unit, and as beta unit is coupled with it, there occur some changes in the beta unit as well. One more point to tell you is that beta unit consisting of some tyrosine residues inside the cell. So some tyrosine residues are present on the beta or linked to the beta unit inside the cell. So when the insulin binds, these tyrosine residues get phosphorylated. As there is some activity of tyrosine kinase in the intracellular part of the beta subunit, tyrosine kinase. So that leads to phosphorylation of not only tyrosine but also some IRS molecules, that are the insulin receptor substrate molecules. So they are also phosphorylated. So tyrosine residues are also phosphorylated, and insulin receptor substrate are also phosphorylated. So this activates some protein kinases. And I think you might be all knowing protein kinases are very important for activation or deactivation of some enzymes. So this can lead to biological changes. So this was the one way through which the receptors of insulin can act, but there are some other 2 ways. So in total there are 3 ways through which insulin receptor can do its function after the insulin binds. First one was the signal transduction that we discussed. The other 2 are the insulin mediated glucose transfer or glucose transport, and third is the insulin mediated, I have read just M, in your notes you are at full, insulin mediated enzyme synthesis. So signal transduction means the binding of insulin makes some conformational changes in the beta unit, which leads to phosphorylation of the which leads to activation of the tyrosine kinase, which phosphorylates the insulin receptor substrate, which in turn activates the protein kinases, and which leads to some biological changes. So this was about the signal transduction. What is insulin mediated glucose transport? The binding of insulin also causes the translocation of vesicles present in the cell. So there, suppose this is the cell membrane. So this is cell membrane. So inside the cell there are some vesicles which contain glucose transporters. So what happens when insulin binds, It causes the translocation of these glucose transporters to outside the cell membrane, which leads to more intake of insulin which leads to the insulin mediated uptake of glucose by the cell. So this causes insulin mediated glucose uptake. So in this, there is some conformation change activates the or translocates the vesicles containing the glucose transporters. The majority, there are many glucose transporter 1 to 7, but the insulin mediated glucose transporter is GLUT4. So insulin causes translocation of the GLUT4 from inside the cell to the surface. Hence, it mediates the glucose uptake by the cell. Moreover, insulin also causes synthesis of enzymes called such as glucokinase, phosphofructokinase, pyruvate kinase, etc., which are responsible for the various metabolic action of these enzymes. So insulin can act by the signal transduction and creates the protein kinase, leading to biological changes. It also leads to the insulin mediated glucose transport or glucose intake by the cell, and the enzyme synthesis also the function of the insulin. So now let's see what are these biological changes that take place due to the insulin binding to the cell membrane. Action of insulin we're discussing under the 4 headings, that is the carbohydrate metabolism, action on protein metabolism, action on lipid metabolism, and action on growth. So first let's complete about the action on growth. See, as you all know, growth hormone is responsible for the growth, but it has been found in certain studies that growth hormone cannot alone can help in the growth of the body. It needs the help of the insulin. So insulin helps in general growth of the body, so it aids the growth hormone to help in the general growth of the body. So it is due to its action on the anabolism of proteins. So proteins are the building block of the body, so it helps in the building of the proteins molecules, so it also helps in the growth. Now let's start with the action on carbohydrate metabolism. I think all might be knowing the sole action of insulin is to decrease the blood glucose level. So in order to decrease the blood glucose level, it acts on various carbohydrate metabolism actions. So I think various topics that you read under the carbohydrate metabolism are the glycolysis, gluconeogenesis. So meanwhile I am writing all these important metabolic pathways. You also write down in a copy the important metabolic pathways in the carbohydrate metabolism, and we'll discuss how it acts on the different carbohydrate metabolism pathways. So I've written down the various metabolic pathways in both carbohydrate, protein, and lipid metabolism. So in the carbohydrate metabolism, we have glycolysis, gluconeogenesis, glycogenolysis, and HMP shunt. So in order to decrease the blood glucose level, it will inhibit gluconeogenesis and glycogenolysis. Other all will be increased. So only 2 metabolic pathways in the carbohydrate metabolism are decreased, that are the gluconeogenesis and glycogenolysis, which will increase the blood glucose level, but the action of the insulin is opposite to it. So it will inhibit these two metabolic pathways and promote the uptake of glucose by the cell and the utilization. In the protein metabolism, its function is to increase protein. So it will promote protein synthesis and it will decrease protein degradation or protein utilization. In lipid metabolism, its function is to produce more triglyceride and fatty acid, so increase lipid. So it will promote lipogenesis and it will inhibit or decrease lipolysis. So this was the action of the insulin in various metabolic pathways and the growth. So growth is mediated by the protein, action on protein, as it helps in the formation of protein. So this will indirectly helps in the growth of the body. and the clinical significance is the decrease of the insulin or its inefficiency can cause diabetes mellitus that we will discuss in a separate video. So if you like the video, please hit the like button and do subscribe to our channel. Thanks for watching.